Okay, so for us to discuss, or let's say for us to determine the fifth aim of research, we will present to you the concept of osmosis. Osmosis. So when we say osmosis, it is the movement of water, or H2O, from a higher concentration to lower concentration. So meaning to say, it is said that osmosis can affect the size of the cell. Okay? And osmosis can be observed mainly observed in red blood cells, or RBC. Okay? So, in order for us to discuss the fifth aim of research, we will discuss the concept of osmosis, whereas it states that the movement of water from a higher concentration to lower concentration. So, in this case, we will have three setups. Let us first discuss the first setup. Okay? So, in setup A, okay, so in setup A, suppose that the cell, this is the cell, the red blood cell, is composed of 50% water and 50% salt. So salt and ACl, sodium chloride. Okay? And then this cell was actually submerged or surrounded in an aqueous solution. Okay, it is submerged in aqueous solution. And then this aqueous solution consists of 25% water and 75% salt and ACl. So, meaning to say, the tendency of the water, okay, as stated in osmosis, the water or osmosis is the movement of water from higher concentration to lower concentration. So the tendency of water from the cell is to move outside the cell. So meaning to say the 50% water from the cell will now be deducted to only 25%. Okay? It will now be deducted to 25%. Now, the solution, okay, or this solution present around the cell will be known as hypertonic solution. So, hypertonic solution is a solution in which, okay, hypertonic solution is a solution in which there are more solute or salt, okay, outside the region of a particular cell. So we can see that the salt here is much higher than inside the cell. So meaning to say, when the water moves outside the cell, what will happen to the cell? Okay, so try to copy this. So when the water moved outside the cell, meaning to say the cell will become dehydrated. Okay, it will become dehydrated. Then the cell will shrink. Since osmosis can affect the size of the cell, with hypertonic solution, the cell would shrink. It will become smaller. And meaning to say, when the cell becomes smaller, it will die. Okay? When the cell is dehydrated, it will die. So, where can we apply this kind of osmosis? So, okay. This hypertonic solution is used to cure tonsillitis. Okay? Kasi pansinin mo, kapag may tonsillitis ka, ang sasabihin sa'yo ay uminom ka ng or magmumog ka ng tubig na may asin. Diba? And asin is this, NaCl. Why? Because so that the bacterial cell with the virus causing tonsillitis will die of dehydration. Okay? So that is the main reason why we cure tonsillitis with water that is mixed with salt, so that it will dehydrate the cell that causes, or the bacterial cell that causes tonsillitis. Okay? So that would be the first setup of osmosis. Okay? Next, let's go to setup B. Okay. So setup B, suppose that the cell, so this particular cell or red blood cell, it is still composed of 50% water and 50% sodium chloride or salt, NaCl. Then the aqueous solution or the solution outside the cell is now composed of 25%. No, not 25%. 75% of water and 25% of salt. So, which one has the highest concentration of water? Obviously, the solute or the solution, because it has 75% of water. So, what is the tendency of water? The water will move inside the cell. Okay, the water will move inside the cell from higher concentration to lower concentration. So, it will move inside the cell. Now, the solution in which there is less solute, okay, solution with less solute outside the cell will be known as hypotonic solution. Hypotonic solution. Okay, hypotonic solution. So, what will happen if the water came from or the water outside the cell enters the cell? So, the tendency of the cell is to become larger. Okay? There is a tendency that the cell would become larger, and then the tendency of the cell is to burst or rupture. Okay? It will burst. It will become bigger. It will become larger. So that is another concept of osmosis, which says that it can affect the size of the cell. Okay? So, and lastly, we have setup C. So, suppose that in setup C, the cell still composes of 50% water and 50% solution of salt, NaCl. Then the outside of the cell, or the solution outside of the cell, also composes 50% water, H2O, and 50% salt. Now, in this case, we can say that the concentration of water is equal to both the inside and outside of the cell. Okay? So, meaning to say, if there is an equal concentration of water inside and outside of the cell, there will be no changes in the size of the cell. Now, what do you call the solution when the concentration of water and salt is equal to the concentration of water and salt outside the cell? So, this solution will be known as isotonic solution. Okay? Isotonic solution. So, from the prefix iso, okay, iso meaning to say equal or in the middle. Okay? So, it is isotonic solution. Now, presented, okay, with the illustrations of osmosis. Okay? Presented with the illustrations of osmosis. Did we validate its truthfulness? Okay, because osmosis said that it affects the size of the cell. And also it says that water with higher concentration will move to water with lower concentration. So meaning to say, it is a fact. So it is validated. Okay? The fact is validated. So with that said, with the illustrations of osmosis, we can determine now the fifth aim of research. And what is the fifth aim of research? Okay? Can you guess? So the fifth aim of research is to verify or validate something. Okay? The fifth aim is to verify or validate something or to prove that something is true. So with the illustration of osmosis, we have proved that osmosis really affects the size of the cell. Okay? So that would be the main idea for the fifth aim of research. So in our next video presentations, we will now be discussing the sixth aim of research.